Cells need energy every second to stay alive. They need it to move molecules, build proteins, pump ions, and divide. But here's the key idea. Cells cannot use the energy in food directly. Food has energy stored in its chemical bonds, and cells must convert that energy into a usable form called ATP. ATP, or adenosine triphosphate, is the cell's energy currency. When ATP loses its last phosphate, energy is released instantly and can power cellular work. Almost every energy-requiring process in the cell runs on ATP, not on food itself. So how does food become ATP? It happens through a process called cellular respiration. The main goal of cellular respiration is to slowly break down food molecules and capture their energy in ATP instead of losing it as heat. Food molecules, like glucose, contain high-energy electrons, especially in carbon-hydrogen bonds. Cells extract energy by moving those electrons from food to oxygen in many small enzyme-controlled steps. This controlled movement of electrons is where the usable energy comes from. Let's follow glucose. The first stage is glycolysis, which happens in the cytosol. Glycolysis splits one 6-carbon glucose molecule into two 3-carbon molecules called pyruvate. This step does not require oxygen. The cell spends 2 ATP at the start, then makes 4 ATP later for a net gain of 2 ATP. Glycolysis also produces NADH, which carries high-energy electrons. If oxygen is available, pyruvate enters the mitochondrion. Each pyruvate is converted into acetyl-CoA. During this step, carbon is released as carbon dioxide, and more NADH is made. This is the point where carbon from food starts leaving the body as CO2. Next comes the citric acid cycle, also called the Krebs or TCA cycle. This takes place in the mitochondrial matrix. Acetyl-CoA is fully broken down, releasing the remaining carbons as CO2. Very little ATP is made here directly. Instead, this stage loads electron carriers. Large amounts of NADH and FADH2 are produced. These molecules hold most of the energy taken from glucose. Now comes the most important stage, oxidative phosphorylation. This is where most ATP is made. NADH and FADH2 deliver their electrons to the electron transport chain, a series of proteins in the inner mitochondrial membrane. As electrons move through the chain, their energy is used to pump protons across the membrane. This creates a proton gradient, which stores energy. Protons then flow back through an enzyme called ATP synthase. As protons pass through, ATP synthase physically rotates and uses that energy to make ATP from ADP and phosphate. This process is called chemiosmosis. Oxygen plays a critical role here. At the end of the electron transport chain, oxygen accepts electrons and protons to form water. Without oxygen, the chain backs up. NADH cannot unload electrons, and high-yield ATP production stops. In total, one glucose molecule typically produces about 30 to 32 ATP in aerobic respiration. The exact number varies, but the big idea matters more than the number. Most ATP comes from the electron transport chain, not from glycolysis or the citric acid cycle. What if oxygen is not available? Cells use fermentation. Fermentation does not make extra ATP. Its only job is to regenerate NAD+, so glycolysis can continue. The ATP yield stays low at 2 ATP per glucose, which is enough for short-term survival, but not efficient. Cells can also get energy from fats and proteins. Fatty acids are broken into acetyl-CoA and produce lots of NADH and FADH2, which is why fats yield more energy per gram than sugars. Proteins can be used too, but only after their amino groups are removed and their carbon skeletons enter the same pathways as glucose. So the full story is this: cells slowly break down food, move electrons to oxygen, store the released energy as ATP, and use ATP to power life.